Hello friends, I am Zaiba Nishad Banu, Associate Professor and HOD, Department of Botany, Vijaya College, Bangalore. Today, let us study the topic on lichens. Let us study the morphology, structure and reproduction of lichens. This topic is for the second semester BSc CBZ students from the botany portion. Now, what are these lichens? They are a small group of curious plants made up of algal and fungal components living together in an intimate symbiotic relationship. You must have seen small greyish patches on the barks of trees. Sometimes the patches are orange-yellow in color. These patches are nothing but the lichens. These lichens are made up of two components, namely the algal component, which is known as the phycobiont, and the fungal component, which is known as the biont. So both these components, that is the algal and fungal components, together form these lichens, and in the lichen plant body, you can neither distinguish the alga nor the fungi. What I am trying to tell you is both the algae and fungi almost lose their identity. Now these lichens, they have a composite thalloid structure where the algal component performs the function of photosynthesis, produces carbohydrates, whereas the fungal component is responsible for absorption and retention of water. Now these lichens have certain other features also. They reproduce asexually by forming various types of asexual spores. We will come to that a little later. The fungal component alone is involved when it comes to sexual reproduction. The fruiting bodies of lichens are shaped structures called as the apothecia. The growth of lichens is extremely slow. They require high temperature and dry conditions for a very long time. Hence, they can grow easily on dry substratum. Now these lichens, they produce a specific acid which is known as lichenic acid, and you must have already heard that these lichens are the pioneers of vegetation. They are the pioneers of vegetation because with the help of this lichenic acid, they are able to grow on barren rocks. Now let's see the distribution of these lichens. There are about 400 genera of lichens widely distributed in most. They usually grow on barks of trees, logs of woods, bare rocks and other similar situations. You can see these lichens growing on the barks of this tree here. There is a log of wood here, and you can see small cup-shaped structures here, very conspicuous, very tiny cups. These are nothing but those cup-shaped reproductive structures which we term as apothecia. You can also see here that this lichen is orange-yellow in color. One more lichen is there here. You can see it is sort of foliar in nature. This is also growing on the bark of a tree. And some of the common species to name are Cladonia aggregata, Graphis duplicata, Hematoma species, Peltigeria scutata, Phaecia aspera and Usnea aspera. Now here I have some more pictures of lichens. This is also a foliose lichen. This is a fruticose lichen. This is the lichen that we just now mentioned, Usnea. This is also a lichen. Here also there are two colors of lichens here. Here you can see some greyish white masses. This is also a lichen. You can see some orange masses here on this bark of the tree, the log of wood. These are also lichens. So all these lichens, they appear really beautiful to the naked eye. Now let us understand the nature of association of the algal component, which we understood as the phycobiont, and the fungal component, which we called as the mycobiont in lichens. In most lichens, say about 75% of lichens, the algal component are the members of chlorophyceae, and the fungal component mostly belongs to Ascomycetes. Now the interrelationship between the algal and fungal components are usually considered as symbiotic, in which both the algae are mutually benefited. How are they mutually benefited? It is believed that the alga synthesizes organic food material sufficient material so that it is available for the lichen thallus, while in exchange of food, the fungus supplies the water and minerals to the alga. Besides this, the fungal component also protects the alga from high temperature and desiccation. However, some workers also believe that the fungal component has a partially or wholly parasitic relationship with the algal component. Now, how is this? This view gets the support from the fact that the hostoria of the fungi are seen in algal cells of some lichens. And on separation, the algal component of the lichen can live independently, whereas the fungal component cannot survive. So, this has led to the conclusion that the fungal component has a partially or a wholly parasitic relationship with the algal component. There is another view. Now, according to this, the association of both the components of the lichens is symbiotic. The fungal shows free dominance over the algal component, and the algal component is subordinate to the fungal partner. Such an association is known as helotism. Now, to expand this view, I told you already, although the association appears symbiotic, the fungal component shows some kind of a predominance and the algal component becomes a subordinate partner. Hence, because of this kind of relationship, the association is termed as helotism. Now, let us move on to the classification of lichens. Now, these lichens, there is no natural system for the classification of lichens. On the basis of the structure of the fruiting bodies, the fungal component, the lichens are classified into two categories. Ascolichens, example Parmelia. You can see the composite thalloid structure of Parmelia here. Basidiolichens, when the fungal partner belongs to the class Basidiomycetes of fungi, the lichens are called as basidiolichens. Example, Corella and Dictyonema. You can see some Dictyonema species growing here. So, Dictyonema is a basidiolichen. Now, coming to the structure and ecology of lichens. The plant body, as we have understood, is thalloid, irregular in shape, as we have seen in the photographs, grey or grey-green in color, but also in some species, yellow, red, orange or brown in color. As I have shown you in the previous slides, we saw lichens which were orange, orangish brown in color too. Now, depending on the external morphology, the following three types of lichens have been recognized. What are they? There are three categories: crustose lichens, foliose lichens and fruticose lichens. Now, coming to crustose lichens, these are encrusting lichens. Encrusting means they have a crust-like thallus, a very thin thallus, which is adhering to the substratum, sticking to the substratum, very firmly sticking to the substratum even on the corners of the lichens. It is thin, flat and a hard thallus. Example of crustose lichens are graphis and hematoma. You can see the entire encrusting lichen thallus here. It forms a very thin layer, firmly sticking to this rocky substrata here, very thin and a hard thallus. Such type of lichens are called as crustose lichens. Now coming to the second category, foliose lichens. As the word says, they are leaf-like. So these lichens are flat, leaf-like and have a lobed thallus. You can see here, unlike the crustose lichens, you can see the edges are not sticking to the substratum. They are lobed, foliar, and they are attached to the substratum with the help of rhizoid-like structures which we term as rhizines. Example, Parmelia and Physia. Here you can see the lichen which is called Physia. The third category is the fruticose lichens. What are these fruticose lichens? They are shrubby lichens with well-developed shrub-like cylindrical and a branched thallus. You can see the cylindrical nature of the thallus here. You can see the extensive branching. They grow erect or hang from the substratum, and the plant body is attached to the substratum by a basal mucilaginous disc. So you can see examples are Cladonia and Usnea. You can see Cladonia here I had shown you Usnea in the previous slide. So these are the three categories of lichens based on their external morphology. Now let us come to the internal structure of the lichen thallus. Now you can see that the internal structure of an advanced or a mature lichen thallus, vertical section, it shows four distinct regions. Now what are those four distinct regions? You can see here an upper cortex. You can see an algal zone here, very clear, predominant, an algal zone. You can see some loosely arranged hyphae, which third layer called as the medulla, and you can see some lower cortex here where you can see some and from the lower cortex you find elongated structures, root-like structures, which we term as rhizines. Now coming to these four parts, the first one is the upper cortex, which you can see here. It is the outermost, thick, protective zone composed of compact interwoven fungal hyphae. You can see that this layer is completely compact. In some lichens like Parmelia, there may be irregularly arranged breathing pores on the outer surface. What do these breathing pores do? These breathing pores help in gaseous exchange. Now coming to the second layer, which is the algal layer, which occurs just below the upper cortex. It is also known as the gonidial layer. Why gonidial layer? It is supposed to be, these are the cells which are supposed to help in the reproduction of the algae before it lost its identity to form the lichen thallus. The algal cells remain embedded in between the tangled network of loosely interwoven fungal hyphae. As you can see here, they are there in the loosely interwoven network of the fungal hyphae. In some species like Collema, the algal cells and the fungal hyphae are distributed more or less uniformly throughout the thallus. Such species are known as homoisomerous. The diagram that I have put here is of a different kind of a species, which we term as the heteromerous species, because you have distinct algal and fungal zones. In a homoisomerous species, the algal cells and fungal hyphae together in this entire region, as I am showing you cursor, there is a more or less homogeneous distribution of the algal cells along with the fungal hyphae. You don't find a demarcated algal zone and a demarcated zone of the fungal hyphae termed as the medulla. Now, what you are seeing in the diagram here is in species like Parmelia, where the algal cells have formed a distinct layer. See here, the layer is very distinct. You can see circular cells. We call this distinct layer. And such species of lichens are known as heteromerous. Some common green algae found in the lichens are the species of Protococcus and Cladophora. These are green algae. And the blue-green algae that are commonly found are Nostoc, Glyocapsa and Rivularia. Now coming to the next zone, that is the medulla, it is the central part of the thallus composed of loosely woven fungal hyphae with large spaces in between them. Yes, you can see the spaces here. The fungal hyphae are loosely arranged. Coming to the last layer, which is termed as the lower cortex, this zone is composed of compactly arranged hyphae which lie parallel or perpendicular to the surface of the thallus. Some of these hyphae become specialized, extend downwards from the lower surface of the cortex, attachment of the thallus to the substratum. These hyphae are known as rhizines. So I think with this we have clearly understood the internal structure of a lichen thallus. Now moving on to reproduction in lichens. They reproduce both by asexual and sexual means. What are the most common methods of asexual reproduction? The most common method of asexual reproduction is fragmentation. Now as you know, the word fragmentation is self-explanatory. Small fragments of the thallus that are formed by accidental breaking or by the death and decay of the old parts of the thallus just separate from one another, and each fragment develops into a new thallus provided it contains both the algal and fungal components. So this is about fragmentation. Next type of asexual reproduction is by the formation of soridia. Soridium is singular, soridia is plural. These soridia, they are small bud-like outgrowths with one or few algal cells. You can see here, there is just one algal cell and some fungal hyphae. So this whole structure is a psoridium. They form a granular layer of greyish-white color on the surface of the thallus. They get detached by the action of wind or rain, and on getting a suitable substratum, they form a new thallus. So this is how the lichens reproduce by the formation of psoridia. Next type of asexual reproduction is by the formation of cephalodium. Cephalodium here, this is the thallus. You can see the cephalodium here. They are small wart-like structures. Small wart-like structures mean small irregular outgrowths formed on the surface of the thallus. And what is the characteristic feature of a cephalodium? The characteristic feature is that its algal and fungal components differ from that of the thallus it is growing. Why is this so? It is due to the fact that cephalodia develop on younger parts of the thallus from psoridia of some other species. Just now we understood what psoridia are in the previous slide. They are a circular mass of one or two algal cells wrapped in some fungal hyphae. And on detachment by wind and rain and finding suitable substratum, they grow into new lichens. So here what has happened is, accidentally the detached psoridia of one lichen thallus has fallen on a young thallus of a different lichen. And since it has got a suitable substratum, the cephalodium are growing. So in nature, you find such close association of lichens from different algal and fungal components. Next type of asexual reproduction is by the formation of isidia. Isidia plural, isidium singular. Now what are these isidia? They are small stalked greyish black coral-like outgrowths shaped like a coral which develop on the upper surface of the thallus. The isidium, you can see the isidium here. They have an outer cortical layer. You can see one cortical layer here, enclosing the algal and fungal components. It is usually constricted at the base. Constricted means it's narrow at the base, and it is easily detachable from the parent thallus. So once it is detached from the parent thallus, it develops into a new lichen. Now with completing asexual reproduction, let us move on to sexual reproduction. In lichens, the process of sexual reproduction, as we discussed earlier, is performed only by the fungal component, which mostly belongs to the class Ascomycetes. So sexual reproduction follows the same pattern as we see in Ascomycetes fungi. The female sex organ is known as the carpogonium. Now this carpogonium is differentiated into a basal coiled region called ascogonium and an elongated multicellular trichogyne. The basal ascogonium is embedded in the algal layer of the thallus, whereas the trichogyne projects over the surface of the thallus. The male sex organ is a flask-shaped structure called spermogonia, which produces the spermatia or the male gametes. Now the spermogonium usually develops close to the carpogonium, enabling the spermatia to adhere to the projected sticky trichogyne. So this enables easy fusion of the male and female nuclei. Now once the male and female nuclei fuse, several branched ascogenous hyphae develop from the base of the fertilized ascogonium. The terminal or penultimate binucleate cell of the ascogenous hyphae develops into an ascus. The diploid nucleus then divides first meiotically, so 4 haploid nuclei are formed, and then mitotically to form 8 haploid daughter nuclei. Each haploid nucleus metamorphoses into an ascospore. So you can actually see the vertical section of the apothecium here. You can see the fertile layer here. You find several asci. You can see here, these are the elongated structures called asci, which bear the ascospores. These asci are intermingled with sterile hyphae. You can see these elongated sterile hyphae here. These sterile hyphae are called as paraphysis. You can see these a cluster of apothecia here, small cup-shaped structures. You can see the vertical section of the apothecium where you can find the outer layer of the apothecium. You can find the algal layer here. You can see these rounded cells which form the algal layer. You can see the central part of the apothecium which is made up of fungal hyphae. Now here you see the fertile layer. This fertile layer asci, and the asci contain the ascospores which we just now understood. Asci remain enveloped by paraphysis. The somatic tissue surrounding the asci and paraphysis forms the fruiting body termed as the apothecium. So this entire structure, as I am moving the cursor, is the apothecia, which are these cup-shaped fruiting bodies. The ascospores, they vary considerably in their size, shape, structure and septation. Ascospores on liberation produces a new hypha, and when this hypha comes in contact with a suitable alga, it forms a new lichen thallus. Now you can see here I have just put a picture of a scanning electron micrograph of a lichen. You can see big cup-shaped fruiting bodies. These cup-shaped fruiting bodies are called as the apothecia. Now these ascospores, they vary, I told you, considerably in size. Generally, the ascospores of these lichens, they show septation. So you can always see them as some bicelled ascospores. So these ascospores, as I already told you, on liberation develop into new thalli of lichens.